Good morning, everybody. Welcome back to daily lectures at New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We're dealing with the subject of molecular biotechnology, and we're on the subject topic of directed mutagenesis and protein engineering. And the topic would be covered with specific titles that would be ensuing as I proceed with the lectures. Please take notes and adequate screenshots to help yourselves. Welcome all those preparing to appear for the NEET exams, the All India Institute of Medical Sciences and the Council of Scientific and Industrial Research exams to be conducted by the National Eligibility Test Forum. And I wish you all the best. Please do not commercialize any aspect of this lecture or any videos that will be put for your references. Please use them to the best of your preparations to help you pass the exams and help build up a good trivia. Right, welcome back to the subject matter proper. Now let me just go ahead with the details. Well, just an intro before I can go into the details. It's possible with recombinant DNA technology to isolate the gene for complementary DNA, cDNA, for any protein that exists in nature, to express it in a specific host organism and to produce a purified product that can be used commercially. However, the physical and chemical properties of these naturally occurring proteins are often not well suited to an industrial application. In some instances, a protein that is better suited to a particular task may be obtained, and by using a gene from an organism that grows in an unusual, often extreme environment. For example, when an enzyme such as alpha-amylase was required to function at a high temperature, the gene for that enzyme was isolated from Bacillus stearothermophilus, an organism whose natural niche is 90 degrees centigrade at hot spring. In this case, the clone gene produced alpha-amylase that was stable at high temperatures used in the industrial production of alcohol from starch. In addition to isolating natural genes that encode proteins with useful properties, conventional mutagenesis and selection schemes can be used in an attempt to create and perpetuate a mutant form of a gene that encodes a protein with desired properties. However, the number of mutant proteins, each with a different amino acid change, that are possible after the alteration of individual nucleotides within a structural gene by conventional mutagenesis is extremely large. In practice, the mutagenesis selection strategy rarely results in any significant beneficial changes to the targeted protein because most amino acid changes decrease the activity of an enzyme. By using a set of techniques that change specific amino acids encoded by a clone gene, proteins with properties that are better suited than those of naturally occurring counterparts can be created for therapeutic and industrial applications. Such direct evolution of genes encoding proteins of interest has emerged as a key technology to generate proteins with new and improved properties. For example, by altering both the Michaelis constant Km, which reflects the tightness of substrate binding to the enzyme, and the maximal rate of conversion of the substrate into product under defined conditions, Vmax, for an enzyme catalyzed reaction to improve the overall. Well, these are details that I've discussed in my former lectures on chemistry and cellular biology for the NEET exams. I did give some, and the CSI exams. These were discussed as part of chemistry too, right? So go back to my lectures and you should be able to recover these portions. Catalytic efficiency, Vmax per km of the reaction, where Vmax equals the total amount of enzyme present, E0 times the catalytic rate constant, K cat, by changing the thermal tolerance, pH stability of both of a protein, enabling the altered protein to be used under conditions that would inactivate the native version by modifying the reactivity of an enzyme in non-aqueous solvents so that chemical reactions can be catalyzed under non-physiological conditions, by changing an enzyme so that a cofactor is no longer required for certain continuous industrial production processes in which the cofactor must be applied on a regular basis, by modifying the substrate binding site of an enzyme to increase its specificity, thereby decreasing the extent of undesirable side reactions, by increasing the resistance of a protein to cellular proteases, which simplifies purification and increases the recoverable yield, by altering the allosteric regulation of an enzyme to diminish the impact of metabolic feedback inhibition and increase the product yield, and to directed mutagenesis procedures. It is not a simple matter to produce a new protein with specified predetermined properties. However, it is quite feasible to modify the existing properties of known proteins. Theoretically, these changes can be carried out at either the protein or the gene level. However, chemical modifications of proteins generally are harsh, non-specific, and required repeatedly for each batch of proteins, so it is preferable to manipulate the DNA sequence of a cloned gene to create an altered protein with novel properties. Unfortunately, it is not always possible to know in advance which individual amino acids or short sequences of amino acids contribute to a particular physical, kinetic, or chemical property. For example, a particular property of a protein may be the consequence of two or more amino acid residues that are far apart from each other in the linear sequence but are juxtaposed as a result of the folding of the protein. In this case, two or more amino acid residues may have to be changed to produce a protein with desired properties. In the not-too-distant future, computer programs may be able to make accurate predictions of protein function on the basis of deduced amino acid sequences, thereby simplifying the task of producing a protein with specific predetermined properties. At present, although it is relatively straightforward to introduce new coding information into cloned genes, large numbers of novel proteins must often be assayed to determine whether a particular property has been created. The process for generating amino acid coding changes at the DNA level is called directed mutagenesis, determining which amino acids of a protein should be changed, and to attain a particular specific property is much easier if the three-dimensional structure of the protein or a similar protein has been well characterized by X-ray crystallographic analysis and other analytical procedures. However, for many properties, such detailed information is often lacking, so directed mutagenesis becomes a trial and error strategy in which changes are made to those nucleotides that are most likely to yield a particular change in a protein property. Then, of course, the protein encoded by each mutated gene has to be tested to ascertain whether the mutagenesis process has indeed generated the desired change. A large number of experimental approaches have been devised for the directed mutagenesis of clone genes. Some of the methods are designed to mutate, delete, or add specific nucleotides of a clone gene. Others introduce mutations randomly over a short segment of the clone gene. Okay, we are on directed mutagenesis procedures, and unfortunately, or before that, theoretically, these changes can be carried out at either the protein or the gene level. However, chemical modifications of proteins generally are harsh, non-specific, and required repeatedly for each batch of proteins, so it is preferable to manipulate the DNA sequence of a cloned gene to create an altered protein with novel properties. Unfortunately, it is not always possible to know in advance which individual amino acids or short sequences of amino acids contribute to a particular physical, kinetic, or chemical property. For example, a particular property of a protein may be the consequence of two or more amino acid residues that are far apart from each other in the linear sequence but are juxtaposed as a result of the folding of the protein. In this case, 2 or more amino acids residues have to be changed to produce a protein with desired properties. In the not-too-distant future, computer programs may be able to make accurate predictions of protein function on the basis of reduced amino acid sequences, thereby simplifying the task of producing a protein with specific predetermined properties at present, although it is relatively straightforward to introduce new coding information into clone genes. Large number of novel proteins must often be assayed to determine whether a particular property has been created. The process for generating amino acid coding changes at the DNA level is called directed mutagenesis. Determining which amino acids or proteins should be changed to attain a specific property is much easier if the three-dimensional structure of the protein or similar protein has been well characterized by X-ray crystallographic analysis and other analytical procedures. However, for many proteins, such detailed information is often lacking, so directed mutagenesis becomes a trial and error strategy in which changes are made to those nucleotides that are most likely to yield a particular change in a protein property. Then, of course, the protein encoded by each mutated gene has to be tested to ascertain whether the mutagenesis process has indeed generated the desired change. A large number of experimental approaches have been devised for the directed mutagenesis of clone genes. Some of the methods are designed to mutate, delete, or add specific nucleotides of a clone gene. Others introduce mutations randomly over a short period of the clone gene, thereby creating a panel of mutated proteins among which one or more may have the desired activity. Oligonucleotide directed mutagenesis with M13 DNA.